Hi, welcome to the fourth annual Neuroscience Innovation Forum. In particular, number of topics to cover with an esteemed set of panelists. But before we go ahead and get started, I want to draw your attention to the fact that April, which is this month, is the Parkinson's Awareness Month. So this panel is very timely. And thank you for those listening, many of you who are increasing the awareness of this disease and sharing the resources available to help those suffering from Parkinson's disease cope with their situations as we look for better treatments in this space. So thank you to all. And again, a reminder that we all are in this together, working to help those individuals who are affected by this disease. So again, reminding everybody that April is the Parkinson's Awareness Month. With that, let me go to the next slide. Trying to see if I can. All right, I think it's turning the next slide here. So this panel today. Few words about myself and then hand it over to my co-chair. So I have a responsibility at AbbVie for neuroscience search and evaluation. AbbVie, as you know, is a global pharmaceutical company headquartered in Chicago, but certainly we have facilities globally. Neuroscience is a key area for the company, and certainly we are deeply invested in that space, both in neurodegeneration as well as neuropsychiatry and migraine. With that, let me pass it on to my co-chair, Sumant Kulkarni, for a brief introduction, and then we'll introduce the rest of the panelists. Sumant.
Thanks, Murali, and hi, everyone. My name is Sumant Kulkarni, and I'm a Managing Director and Senior Biotech Equity Research Analyst at Canaccord Genuity. My current coverage is focused on stocks of neuroscience companies, and I keep track of all the events in the neurospace, regardless of whether it's industry-driven by private, public, or nonprofits, to research coming out of academia. So I'm very pleased to be co-moderating today's panel on Parkinson's and movement disorders. I'd like to thank our host, Sachs, for inviting us, and to my co-moderator, Murali, as well as all of our panel participants and everyone that's tuned into this video webcast. All of us know the unmet need in these indications, and there are brave organizations that are trying to help us overcome the problems that these disorders present, but success remains quite elusive so far. We'll be keeping this session very interactive, and we'll start off first with a brief introduction from our panelists and then move on to what we will sure will be a very lively discussion. So if anyone in the audience has any questions, please feel free to send them across using the designated systems interface. We'll weave those into the discussion. With that, I'll hand it off to each panel participant to briefly introduce themselves. We'll go first name alphabetically. And that means Kees is first.
Thank you, Sumant. Thank you, Murali. Hello, everyone. My name is Kees Bain. I'm CEO of BIAL Biotech. So before that, we were actually known as lysosomal Therapeutics. We were just recently acquired by BIAL, a Portuguese pharma company. And we are continuing to focus on genetic subpopulations of Parkinson's disease that are by now validated as targets that increase risk of getting Parkinson's disease, and in some cases actually contribute to faster progression when people actually get the Parkinson's disease. So we focus on several genetic targets that generate enzymes that are functioning in the lysosome, where glycosylglycolipids are broken down into building blocks for glycosylglycolipids that are major constituents of the membrane. And we believe that by doing so, overcoming the enzyme deficiency due to these particular mutations normalizes the glycosylglycolipid metabolism, and with that, the proper functioning of membranes in the brain. Our lead molecule is a glucocerebrosidase or GCase activator that normalizes the enzyme which is impaired due to this GBA1 mutation which was originally found in Gaucher disease, and this molecule is we're preparing for a phase 2 start by next year. Thank you.
Hi, everybody. Marco Baptista. I'm Vice President of Research Programs at the Michael J. Fox Foundation for Parkinson's Research. We're a facilitator and a funder of Parkinson's research, funding now over $1 billion in research programs since we started back in 2000. We don't have any endowment, so we have a very aggressive strategy in trying to raise funds and then getting the funds out to the best Parkinson's research science out there. So it's a pleasure to join this panel, and very happy to contribute to this discussion.
Hi, everybody. This is Milton Warner. I'm the president and CEO of Inhibikase Therapeutics. We're a protein kinase inhibitor-focused company, and we are presently in the clinic with our lead asset, IKT-14809, which has been shown to both halt and reverse functional loss inside and outside of the brain in validated animal models. And we're presently preparing to enter late phase 1, early phase 2 study in Parkinson's patients.
Hi, everybody. I'm Behrouz. Thanks, Murali and Sumant, for having me on the panel. It's really nice to see all your faces again. I'm a neuroscientist by training. I've been focusing on Parkinson's disease for the past 15, 20 years. I've co-founded 2 companies, NI and Vincere, both of which are actively growing and quite different, so I'm happy to talk about either one. NI is focused on computational approaches, particularly trying to both understand the disease and any way that we can accelerate drug development. That includes multiple stages, ranging from using AI for target ID, validating targets using biological simulation, virtual screening of mitochondria, small molecules that we have at Vincere. And as you probably know, there are no clinical trials that are aimed at mitochondrial quality control mechanisms for Parkinson's disease. So us and a handful of other companies are looking to fill this gap. We are early stage, but we're excited to progress our compounds towards the clinic for patients.
Let's go to Tim.
Did I cut out?
Tim, you're on mute.
Thanks. So hello, everyone. I'm Tim Dyer, co-founder, CEO of Addex Therapeutics. Addex is a Swiss public company that recently dual listed on the NASDAQ last year. We were founded around very interesting technologies for the discovery of allosteric modulators. So we've been pioneering this field for 18 years now. And like a lot of biotechs with technology platforms, we had to focus, and we decided to focus on neurology. And our lead asset is for Parkinson's dyskinesia. We have a number of other assets also, so 3 at the clinical stage. The second program is a reformulation of the lead asset for dystonia, which is another movement disorder. The third clinical asset is partnered with J&J and is about to start a phase 2 in epilepsy. And then we have 6 preclinical programs, of which one of them is the mGluR4 positive modulators. And this is an interesting target for Parkinson's disease. And the mGluR2 negative modulator for cognition, which is another target which could be focused at the cognitive impairment that's seen in Parkinson's patients as the neurodegeneration progresses. Very happy to be on the panel. Look forward to the discussion. Thank you.
Thanks, Tim. You can click to the next slide. Yep. Got it. All right, we're going to jump right in today, and what we'd like to cover in the next 45 minutes or so, perhaps 15 minutes each for each topic. We'll first talk about targets and approaches for Parkinson's disease. Obviously, we could have a number of modalities, small molecules, biologics, or other sorts of approaches, but finding compelling targets still remains a key challenge in this field. Not unique to this particular field, but in any area of discovery. And there are different approaches to find and identify those compelling targets. So we're going to spend some time talking about different ways to go about it. Then we'll switch to the next topic, which is around translation and drug development considerations. Obviously, there have been significant advances, but there also remains to be the investment landscape, and several of you who are listening today, are thinking about investments in the space, and certainly we'll cover that as well. Go through what are the recent trends with respect to investments, where are the investment dollars going into, and what are the key challenges for investments. And for smaller companies, of course, partnerships with big pharma or other entities. So those are the three topics we'll cover today. And we'll jump right into the first topic, which is around targets and approaches for the disease. So let's flip to the next slide if I can. There's a delay between slides, so okay, here it is. So why don't we start today here? And as I mentioned before, identifying compelling targets remain a challenge, and we're going to open up with understanding what are some of the key considerations with respect to selecting the right target. In your introduction, you did mention the approach you're taking with respect to genetically defined targets is sort of the starting place for the discovery efforts. And maybe you can elaborate that. Not only from the context of how we think about GKs and the biology, but even other targets where that sort of an approach is sort of the prioritized way to go about. So, Kees, do you want to kick off that conversation?
Yeah, sure. I mean, you already heard quite a portfolio of targets from the introductions of the panel members, all the way from symptomatic agents to putative disease-modifying agents. Or targets in the mitochondria. We are focused on those targets that occur in the lysosome and that have to do with what I mentioned, the glycosphingolipid metabolism. And in genetic research done across the world, it's now become pretty clear that some of these targets have now been, if you will, validated, at least as risk factors. The most prominent and probably the most described one is the GBA1 mutation, which of course is also in a dual allele mutation, the cause of Gaucher disease. But if you're a carrier, you clearly have a higher risk of getting Parkinson's disease. And what is now becoming more and more clear also is that as carriers, when you have or get the Parkinson's disease, you actually will progress faster. So this is for us the major topic of research. We believe it offers a wide range of subsegments. So our approach, of course, is to go after each of these genetic subsegments. With specific, in most cases, agonists of agonizing or normalizing enzyme function to normalize the glycosphingolipid metabolism. So it's a very personalized approach, very specific, and it adds up to the GBA1 mutation occurs in roughly 10 to 15% of Parkinson's patients. It's higher mark penetration in Ashkenazi Jewish population. So, of course, the attractiveness of this is that it is clearly associated with as a risk. Now, the issues, of course, are that if you have this mutation, your risk of getting Parkinson's is increased, but up to about 10 to 15-fold depending on the severity of these mutations. So if the average risk is about 1% at 65 years old to get the disease, it means that even with the most severe mutations, your risk goes up to about 15%. So something is interesting going on that still, you have a much higher probability, even with the mutation, to not get Parkinson's disease, or not at the average of 65. So we'll probably talk about this at some point, because as interesting as these genetic targets are, and as, if you will, more and more becomes clear as to what causes these Parkinson's disease, I think what is equally important and may deserve some discussion is why are still most patients if truly protected against these mutations. And I think that it is, whatever constitutes protection through other and so-called suppressor genes, etc., they probably are as or maybe even more interesting targets. But that's really that is in its infancy. But this is definitely something that I think should require a lot of attention as we go forward.
Agreed. And again, it's not a linear path as well. I think it sometimes tends to be a vicious cycle, not only involving the lysosomes, but certainly other organelles that are equally important. And, speaking of organelles, I think mitochondria is another important space where, again, there are interesting clues from genetics. But maybe, Spring, if you want to elaborate on how you think about targets in the mitochondria, and perhaps take us into a conversation around what you touched at your introduction, how you're utilizing AI-based approaches to, distill and have a different perspective on target identification.
Yeah, absolutely. And I agree with Kees. We think about the genetics very, very much as well. I think the genetics give us more of a cause and effect relationship, in the sense that we actually know these particular proteins contribute to the disease. And on the protection side as well, although, as Kees mentioned, much less has been done on that front, it'll be very interesting once we have more information on that. We do use a lot of computational approaches. I think that it's becoming essential with the vast amount of data that's available to us. Particularly, we can use human data because models of the disease are not great, as I think we're all aware. For target ID, we've used an array of AI approaches, but one that's been really fruitful for us and fits into this topic of genetics is we looked at the interactome of genes that we know cause PD, and we know that a lot of these genes seem to work through separate mechanisms. So, LRRK2 and trafficking, GBA and lysosomal function, Parkin and mitochondria, for example. But all of these converge on neurodegeneration and manifest in a Parkinsonian phenotype. So we sort of asked the question of, is there a mechanistic point of convergence? And using the computational approaches, we were able to narrow in on mitochondrial quality control mechanisms as the strongest point of convergence that we see between these genes that all contribute to Parkinson's disease. So, and really in the recent years, since we've done this work, which was quite a few years back, we've now seen multiple groups showing that there are deficits in mitophagy in cells, for example, from patients that have mutations in these other genes, and also animal models, for example, GBA, LRRK2, synuclein even, where mitochondria are not considered to be the primary mechanism of action, we're still seeing these deficits. So, we really think about these as ways of nominating these new targets, and I think there are some caveats as well. Some of the targets are well known, like Parkin, not new. Some are just emerging, like USP30, and some have just not been examined in Parkinson's disease at all. So we really have our work cut out for us to show that these are actually relevant in the etiology of Parkinson's disease. But I think it's worthwhile because, particularly if you can stay close to the disease by examining human patients with Parkinson's disease, I think it's a worthwhile approach to take.
Great. And Milton, would you like to add to that, speaking of the point of convergence, because you think about convergence in a slightly different way as you think about biochemical pathways as well. You'd like to clarify and maybe add a few more points that will help.
Yeah, that's right. I think Inhibikase has taken an orthogonal view to what has been traditionally been viewed as trying to elucidate information from clinical studies or from clinical projections. And Inhibikase's process has really been focused on elucidating the biochemical pathways in their entirety And we have found that newly developed animal models for which we played a part in their development actually faithfully reproduce many of the early steps and allow one to manipulate different points in the pathways and see the right biochemical outcomes, the right physiological outcomes that are reflected in patient symptoms. So I think that's opened up, for us at least, an entirely new set of targets that were not previously considered. How does one manipulate what's going on in the disease process? Because the fundamentals of that disease process are now largely known, still again derived from validated animal models, but they're beginning to be now validated in patients. And so I think that approach, which I think has traditionally been a very good approach in elucidating targets and developing drugs against them, particularly against enzymes, where the industry has been most successful in developing pharmaceuticals, really comes to fruition. And that's what we've seen in our approaches.
Right, understood. Maybe just to follow up on the topic of early targets, and Marco, you see a number of early targets, as folks approach your organization. You also see a number of early modalities as well, right? Beyond the small molecules or the biologics approaches, other modalities that are being explored. Would you be willing to sort of comment on your perspective with respect to both targets and modalities in the way things are evolving today?
Yeah, in general, at the Fox Foundation, we're trying to create a very broad portfolio because we understand how complex the disease is and how heterogeneous it is. And there's a lot of interest around. We've made a lot of investments around the genetic targets, primarily because we don't really know the cause of most Parkinson's cases, and that's why we classify them as idiopathic. So we feel it's a good starting point, but the end goal is not to just be trying to find treatments for the minority of individuals that are different mutation carriers. It's to try to better understand if these pathways are pathological in idiopathic Parkinson's patients. So that's been a general approach, one in which we're trying to be very holistic. We're trying to be very broad. With regards to modalities, we're always looking to try to pivot our strategy so that if it seems like there's been some good successes preclinically and in the clinic with one particular type of modality, a small molecule, for example, let's say with LRRK2, then we're looking to see maybe are there other ways to target the LRRK2 protein? Are there other ways of targeting without using small molecules to make sure that we just have as many shots on goal as possible? So we don't have one horse in the game, and we're very open to new ideas, and we're not risk averse. But I think we understand that given the complexity of the disease, we have to keep a very open mind and try a lot of different approaches.
Yeah, good point. And Tim, before we move away from this topic, are there any other points you do like to add? I know you've got programs that are fairly advanced. You're locked in on the targets, but any lessons that you've learned along the way with respect to selecting the right modality, the right targets?
Yeah, I mean, Addex is very much focused on symptomatic treatments, because that's where our allosteric compounds around metabotropic glutamate receptors are focused. We've spent a lot of time trying to understand how glutamate interacts and plays a role within Parkinson's, because everybody believes Parkinson's, which is correct, is a dopaminergic disorder, and the mainstay of therapy today is L-DOPA. But because we are focused on glutamate, we've been looking very much at how glutamate plays a role, and it seems that glutamate and basically the removal of the inhibitory pathways on the glutamatergic tone in certain parts of the brain are what are causing things like dyskinesia. And now that patients are well treated to some extent by L-DOPA, they're living for a lot longer. We're now seeing a lot of these other diseases, which are, the PD psychosis, the agitation, dyskinesia, and a whole host of other symptoms, which are having a major impact on the patient's lives. I think, and I think there are other groups as well that are starting to look at this and seeing this as a very interesting area for investment. It is an area which has been underinvested because the industry has been very much focused on disease modification.
Yep. And we'll talk about investments on the last section. We'll spend much more time on covering that topic. And Sumant, would you like to add a bit more in terms of, as you scour the landscape and look at many other companies, are there any trends that you're seeing in this space with respect to, targets and other approaches here?
Sure. So the trends that I see are mainly driven sometimes by investors, because most investors like to see, target engagement, perhaps a biomarker or two, a specific unmet need, and how well that might be targeted, versus a more, using a more defined genetic approach versus a more amorphous approach to targeting the whole disease. A well thought out clinical plan with the perception that the FDA and eventually payers may be clearly on board. But as we know, this is very difficult science, and investors can't get things right all the time. So I would think diversification would be important, especially when placing investments within the neuro space. So I do think that newer modalities and all that can capture the fancy of investors, but that can blow hot and cold sometimes, as we can see with the gene therapy landscape. For example, we had a recent takeout in this specific space. Prevail is a company that got bought by Lilly, had a Parkinson's program, which was more genetically defined compared to tackling the entire disease. So somewhat like what Marco said, I'd like to echo those comments.
Mm-hmm. Great. All right, I'm looking at our screen for any questions, but I don't see any questions for this particular topic or section. So why don't we, in the interest of time, move forward? And again, just to remind those of you listening in, if there are questions, please go ahead and ask them. We can incorporate them as part of the answers or ask them specifically. Happy to do that. I see one question here. Let me just read it out. Can everybody see it? The question is around thoughts about the toxic forms of alpha-synuclein as a PD target. So the question from Tim is, how do we think about what forms of alpha-synuclein is relevant with respect to targeting as far as PD is concerned? And of course, this is not a topic that we can answer in probably 30 seconds. It's a controversial topic, obviously, but perhaps who would like to take it on? Thoughts about the toxic forms of alpha-syn as we think about PD and targeting.
Well, Murali, we obviously, we're focused in Inhibikase on developing c-Abl kinase inhibitors. We hold the view, which is an emerging view, that the alpha-synuclein form that is toxic is entirely intraneuronal and rarely is seen by the outside world. It moves between neurons through direct contact predominantly, and that's been shown in a variety of different ways. As a consequence, it's difficult to develop a therapy to clear directly by going after the toxic synuclein itself as we've defined it. But what leads to that formation can drive the recovery of intraneuronal clearance mechanisms, either through autophagy and lysosomal degradation or through the proteasome pathway. And both of those have been proven to be successful in validated models to completely clear toxic synuclein from the affected tissue, accompanied by recovery of functionality. So we think it is an accessible topic, but not the direct interaction with the toxic form, or whatever you want to call that form of synuclein itself.
Mm-hmm. Anybody else would like to add anything further to that comment? Or
maybe this case. When you, for instance, I attended the ADPD meeting just recently held, and I was I was surprised that even coverage of what we call new targets, whether it's genetic, mitochondrial, etc., it's always somehow linked back to alpha-synuclein, and so somehow there is this deep-seated notion that alpha-synuclein has something to do with Parkinson's disease, and somehow everybody likes to make the more specific target-based approaches, genetic or other, always relate back to alpha-synuclein because that's what they think, or that's what we're looking for. How do you think about that? Is that the right approach? Because sometimes I wonder that we may be reaching a little bit too far to seek these relationships with alpha-synuclein, but I'd love to get your perspective. Milton.
So I can tell you this pretty directly. So it's very clear that synuclein plays a role in triggering the early steps of the biochemical processes we think are driving the disease. So we don't view synuclein as a source of causation. It's necessary, but it's not sufficient. But what you can show, and you can show this very clearly, that when you trigger the intracellular processes by the affected neurons taking up aggregates of synuclein, we can call them whatever they like, they're chemically modified, they have an altered conformation from even those that we've characterized extracellularly, and those chemically modified forms of synuclein are what are triggering the biochemical processes that lead to both the mitophagy effects, the effects on the ubiquitin proteasome pathway, the loss of lysosomal degradation, the induction of the apoptosis, and prothanatose. And if you do things to undo that process by shutting down the processes associated with chemical modification of synuclein itself within the affected neurons, you see stepwise each of those affected systems reverse. And when you do so, you clear the tissue in a living organism of the presence of that toxic synuclein, and that's accompanied by almost complete functional recovery. And outside of the brain, in the GI tract, where the other major organ of effect is in patients, you don't just see recovery of functionality measured by the ability to properly process and digest food, what we might call the whole-to-gut transit time. You see that within the tissue itself, and you recover normal neural anatomy because you can't induce the stem cell compartment in the ENS, which is constantly regenerating neurons, to build the ENS properly. In the brain, while you can rescue neurons, we don't know presently whether you're seeing regenerative events. Obviously, that would be an unexpected thing based on our paradigms about what happens in the brain. But there clearly is recovery of activity that you can see and rescue functionality.
All right. So do you then believe that to interfere with the formation of toxic alpha-synuclein constituents, to go after these processes that we've been discussing, what do you think about going after synuclein itself with an antibody?
It's proven to be I better choose my words carefully. The recently described results from phase 2 trials confirm what we've seen in other misfolded protein diseases. The pursuit of sequestration of extraneuronal synuclein aggregates in any form that has been at least touched by an antibody appears to be ineffective by any measure that we would use inside or outside of the brain. In Parkinson's, it's been proven to be ineffective in every experiment, perhaps except for one, the Lilly antibody, which is looking at a chemically modified form of amyloid plaque in Alzheimer's. It's proven to be ineffective in ALS. It's proven to be ineffective in FTD. It's proven to be ineffective in Huntington's. So I think while it's appropriate that we have recognized the presence of these misfolded aggregates as being associated with the disease, they arise from the disease. They are not actually causing it, but they are driving the steps that are really at the heart of the biochemistry of the disease process. At least that's where we have gone in Parkinson's. And you see, when you block those events, you see the recovery of functionality along with the clearance of the thing we've been trying to get rid of all along, that is just a phenomenology associated with the disease itself. So I think we're starting to get at the problem of recognizing what is the misfolded protein actually doing. It's not the cause of the disease, but it certainly is required for the process of the disease to progress. Yeah.
Milton, so with that, I think we should probably move to the next topic. And maybe this is really an interesting topic as well. I mean, it's certainly not going to be one mechanism or one approach that we should consider. There could be, you may have to deal with the clearing misfolded proteins. You may have to interfere with other pathways. So it could be, not one magic solution. There could be different ways to effectively stop different aspects of the disease process. So that's really an important point. So why don't we do this? Let's go to topic number 2. And Sumant, if we can maybe jump into our topic on translation and drug development considerations. So I'm sure some of these topics will come back again, but let's spend the next 15 minutes or so on the second topic.
Sure. Thanks, Murali. And we've touched upon this briefly. We'll go into a little more specifics here. So, biomarkers and translational tools remain a very critical aspect. Lots of efforts are being undertaken here to leverage molecular imaging and clinical data So I'd like to pose a question, maybe start with Marco, because he has a bird's eye view of this space. What progress is being made, and what are the key hurdles that must still be overcome on this front?
Yeah, I think probably one of the top priorities at the Fox Foundation is to really better understand biomarkers and things that we can measure to help better diagnose Parkinson's patients, better enrich for clinical trial populations, and just being able to have very clear readouts in the clinic so that we can properly interpret the data. We've been supporting this observational trial, the PPMI, the Parkinson's Progression Markers Initiative, now for some years, which started off looking at newly diagnosed, non-medicated Parkinson's patients with the relative controls, also looking at some genetic cohorts and following them over many years to look at clinical readouts, to look at different types of biological readouts and collections. And, this is a very large effort that takes a lot of participation, not just from Parkinson's patients, but from people who do not have PD and people who are supporting the Parkinson's population, and trying to best understand how we can sort of match these biomarkers with the progression of the disease. We've now are starting to move into sort of a second phase of doing PPMI, and that's trying to look at a prodromal aspect of what could be occurring before an individual is actually showing any of the motor symptoms, and trying to see if there are readouts that we can look at or different molecular signatures that we can see, so that we can be able to predict when a person might then phenoconvert. And so we're now moving into that second stage, where we're going to have a lot of different clinical sites that are going to be open, about a total of around 50. Going to try to incorporate a little bit more about with digital sensors to be able to better get a more objective viewpoint of the symptoms of Parkinson's patients. And in doing so, it's going to require a lot of participation. So we're going to try to do as much work as we can remotely to try to better understand what the patient-reported outcomes are, trying to understand if there are certain issues with regards to REM behavior disorder, for example, or smell tests that can be administered in a remote manner, so that we can start to narrow in more and more on a set population which we are feeling more confident that could be phenoconverting. And so the goal is maybe to start with like 100,000 people with very remote kind of outcome measures, patient-reported outcome measures, and then sort of narrow it down so that we can try to get another 2,000 individuals that are now showing readouts with DAT scans, and then can fully participate in all of the aspects of what this observational study has to offer. So that is a huge effort at the Fox Foundation. Requires a lot of individuals to participate in, but we think there's a lot of value there, because in the end, it's giving us a better understanding of the disease, which is still lacking.
Got it. And then for Kees and Milton, any specifics you might have to offer on target engagement in the brain in this context?
Yeah, it's actually also responding to the comments just made. Is that if you look, for instance, at, in this case, our primary target, GBA1, right? I mean, so the good news is that you can identify these patients because you can genotype them. The issue, of course, is, and what is also known is that GBA1 mutation is a risk factor. But we still don't really truly understand what actually causes the disease if you have the GBA1 mutation, because there are, as any biological system, which is extremely variable. There are a lot of GBA PD patients who have, if there is such a thing as normal levels of GCase activity, they have higher than normal GCase activity. And then there are a lot of idiopathic PD patients who do not carry the mutation, who have lower than normal, right? So, and so how do you? So, and then the question maybe for you also, Marco, is, okay, fine. First of all, you can identify these patients, then you can determine their GCase activity, but it's in the periphery, and ultimate target engagement is to measure effects on the enzyme somehow in the brain, and that takes years to develop, an assay to visualize, in this case, target engagement. And I think that puts us or any small biotech company in a bit of a pickle, right? Because we can either decide to go forward, and the next experiment is just to do it in the human animal, or spend many years trying to come up with a brain penetrant biomarker. And we've decided to go forward with a clinical trial. But I can tell you that in our discussions with pharma, there's been a lot of discussion about, okay, well, we prefer you to actually spend more time on developing a brain penetrant marker of target engagement. So what is your perspective? I mean, how do you make these kinds of trade-off decisions, especially when you think about funding some of these efforts?
Yeah, with regards to funding, I mean, again, I think we keep our portfolio very broad. So we do have efforts where we're really trying to look at biomarkers in the brain. For example, we have a huge effort in trying to find an imaging tracer for alpha-synuclein. I think to your point, I think there can also be a compromise, and there can be work that's done at the preclinical stage where you're really trying to better understand the PK/PD relationship of what's going on in the periphery and what's going on in the brain at the preclinical level, to then extrapolate maybe what could be done in the clinic in the periphery that is potentially a signal of what's going on in the brain. I think that a big part of that can be again seen within the LRRK2 space, where there isn't very clear readouts within the central nervous system of LRRK2 for target engagement, but a very good understanding of the relationship of the periphery to the brain that's been modeled in preclinical models. So I think our perspective has been to try to be pragmatic, try to move as fast as we can, and we have certain projects which are a lot more higher risk than others to try to get at what you were mentioning.
We'll switch it up a bit. I'll get Spring involved here as well on your thoughts, and then we'll turn to Milton because I'm sure everyone has a view on this.
Yeah, we have actually are following kind of a similar to what Marco was talking about with LRRK2, where you're seeing the target engagement in the periphery and then just showing that the compound gets into the brain. I think that's probably as good as it gets. As Kees mentioned, it's much more difficult to try to actually show target engagement in the brain. And it is a spectrum. We see companies kind of ranging from not even really showing target engagement and or not establishing those correlations in preclinical stages of peripheral versus CNS target engagement. And then at least this way, you can have the correlation, and then when you go into human, you can just measure in the periphery. But I think that's probably the sweet spot of where we can pragmatically, I guess, go after the biomarkers of interest, or biomarkers of target engagement. But much further than that, I'm hoping actually, Kees, that we don't have the same feedback from the pharmas that we will be talking to, because part of what we've seen too is it changes, the criteria changes as we improve on the compounds and move forward. So hopefully that's will be sufficient, but that's our approach.
Milton, your take?
Well, so we try to use a combination of some things that are similar to what Behrouz described. We think the GI is an important organ of interest in the global treatment of Parkinson's disease in the body. GI correlates for functionality, for pharmacodynamics, through tissue rectal anal biopsy, et cetera, can be very compelling and combined with the indirect measures in the brain. But we're also exploring, what do you do in the brain? Unfortunately, many of the targets although this group is all pursuing, are low copy number targets in the brain, so you're not likely to see them easily. There have been approaches that were focusing on exosomes, which we've also explored, but we all are aware of the fact that it's hard to tell whether an exosome was really brain-derived to know what you're looking at. So we've become very interested in more traditional spectroscopy approaches, oddly enough. We'd love to use MRI in some way. If you got functional recovery, you could use functional MRI to look at a consequence of a disease treatment. But it's also true that you can do magnetic resonance spectroscopy in the brain, and we're beginning to explore whether or not we can see evidence of enzymological activity through markers that you could introduce into patients safely to be able to make those kind of measurements and really look at direct consequences of your target engagement that way. These are uncharted territories, so no one's going to lead anywhere yet. All of us would love to be able to do PET imaging and get real direct interest of engagement in the brain, but PET, compounds, PET active compounds are unbelievably hard to develop. And I'm not sure the appetite is there to go back to radio tracer studies the way we did 20 or 30 years ago. But in fact, in our view, it shouldn't be ignored.
Right. Now, these are all like super cool things that are going on in the space. But the key question, I guess, is how is this going to be put into practice with ongoing clinical trials? And how is an agency like the FDA going to get on board with these newer types of biomarkers and things like that?
Well, from where we sit, the agency's continuous responses are people having an easier time with life. They're interested in the activities of daily living more so than they are even in improving on falling frequency, on dyskinesia, on off periods, etc. And so I think as we all explore and get into the heart of causation in the disease and therapies begin to impact causation, you're going to see a natural outcome that reflects improvement. None of us know at what point we can no longer find a functional benefit yet. So, and I think most people these days are going after early stage patients where functional rescue is at least formally possible. So I'm personally less worried about the agency than I am about trying to make measurements that are meaningful. And so we've been pursuing it that way. I think the agency, especially in neurology, is very open to all kinds of things because they want to see a broad-based solution and multiple solutions in that.
And this is a very vast topic, so we'll kind of move on to the next one. We can always come back to this, but these is movement disorders and Parkinson's, perhaps uniquely amenable to wearable devices. So in that context, I'll engage Tim here, to talk a little bit about that, dyskinesia, dystonia. How do you expect this to change the course of drug development, clinical practice, maybe actual use going forward?
Yeah, maybe first I'll just touch a little bit on the clinical trial and the patient populations, because it is extremely important to select your patients. And we've, one of our trials didn't do so well because of the patients were less severe than they should have been. So it's very important to make sure you've got the more, especially in the symptomatic treatment of dyskinesia and psychosis and things like this, is that you get more severe patients than moderate. The other thing is Parkinson's has a placebo response, which is physiological because of the underlying dopaminergic nature of the disease. And so this is extremely important. And, that was managing placebo response using a number of techniques. Was very successful in the pimavanserin nuplazid turnaround in PD psychosis, and it's something that we have had to really take into consideration. For example, in the study that's just about to start, we've got a 2-week brief psychosocial therapy BPST process, which really is there to draw out. It's a non-pharmacological intervention, which is there to draw out placebo responses ahead of baseline. So that's just my 2 cents on the patients. But on the wearables, there's been some fantastic progress, even just in the last month, and where we're seeing these sensor-based assessment systems, which are really able to distinguish the normal movement from the abnormal movement. And I think that was one of the really big challenges in the past. We're following that very, very closely. But at the moment, we're in a pivotal study. So, we're a little bit advanced for these sorts of things. But I think we're going to see this becoming a big story in the future. And again, as was mentioned by Milton, activities of daily living and the improvements in activities of daily living is very important to the FDA. And I think what's fantastic about these wearables is they're really able to measure improvements, in the abnormal movement, in these, different activities of daily living, which is again very important for these wearables to get, or these assessment systems to get approval from the FDA.
Got it. So we still have some time left. So I'll go back to that very important question that we had before on the clinical trials part. Anyone else would like to chime in on that? Identification of the right patient population, putting biomarkers into practice in ongoing clinical trials.
I got a question for Tim. I'm wondering, from the wearables, we looked at this question a couple of years ago, and at that time, dealing with the data flow was daunting. It's all, a continuous stream of a lot of data over a long period of time. How do you handle it?
Yeah, I have no idea. I've just been, we're following it.
I see.
And we're seeing a number of papers being published. There was quite an interesting paper just published this month. I don't know how they're managing the data, but I think, what is clear is they've been able to show that their sensor-based assessment system was correlated extremely well with the raters.
I was just going to mention just briefly that within that observational study that I was mentioning, the PPMI study, that there are two digital sensor approaches that are being used that are capturing both passive and active information. It's from the Verily watch and the Roche smartphone, and I totally agree. There's a lot of data that's going to be generated. I think what's really important is to make sure that all this data is then made available to everybody, so that people can then explore it and use different approaches to try to make sense of the data. So I think very soon we'll be able to make available all of that data from the digital sensors that's been accumulated. But yes. Definitely.
So, Mark, I just wanted to also mention so if you look at the wearable results that were used, or the wearables that were used in the Roche, the Pasadena study, anti-amyloid antibody, anti-synuclein antibody, that's very beautiful data. It's very tight, and it shows a nice separation from placebo. So that's something that I don't know which wearable they used, and you say it was a smartphone, but how that worked. But that's definitely something that we should take a look at.
Just going back to the clinical trials biomarkers approach, anyone else would like to chime in on that? If not, we can move on to the next section, which is the investment part. I already made some kind of brief comments on how investors typically tend to approach investing in this space, but I'll start off with actually a question for Murali. Like, you are a representative of a larger pharma company or a biopharma company. Biopharma has generally, the larger ones have shied away from this space. What keeps AbbVie interested from your point of view? Because I think that's very important to know from an investment perspective as well.
Yeah, that's a really good question, and I'm trying to get to the next slide. It's not moving for me, but I know the question is a good question. I think what's keeping us interested in this space is really how the science is evolving in this area, right? When we see the science is moving in a direction that we start to understand the human disease and the biology, that's a place where we think it's primed to take some very calculated risks. Some of these targets are fairly early, but still, if you look at what we've done over the years, we've made really good investments early on. Even seed companies in that stage exploring new areas of biology. All the way through clinical stage assets. So it's really the biology that's evolving and the targets that's coming out of that, and then the enablement that's happened in the field with organizations like the Fox Foundation, really providing that framework for biomarkers, imaging ligands. There is that commonality. Although we may be working in different companies, the questions we're asking related to translation and the clinical trial, there's a lot of commonalities in there. And so the framework that's provided by organizations like the Fox Foundation gives us more incentive to say, well, we don't have to figure it all ourselves. There's a community that's going to be thinking about it. And as long as that framework is available, then, we are open-minded in making the right investments with the right team. So that's kind of the spark that gets us into the space and keeps us motivated to continue to be in the space and really be a player there.
Yeah, thank you, Murali. And I think a big part of what the Fox Foundation wants to do too is just be a neutral convener, so that when there are these questions that the entire field feel are a challenge, and it'd be difficult, or at least it would be much slower to go it alone, to be able to just convene everybody together and then figure out the right mechanism to try to generate the data that's needed to build more confidence around it.
Exactly. And I think there could be common themes. I mean, Kees touched on, target engagement questions, right? I mean, it's a challenge for the field to get to the right target engagement questions. We may be, competing on molecules, but as far as the target and the biology and target engagement is concerned, that could be everybody's problem, and we should work together to tackle it. It doesn't make sense, like, AbbVie making a PET ligand for a target, where, and Merck doing the same thing and somebody else doing the same thing, because that's not what we are competing on. We're competing on the molecule. So, having a PET ligand available for everybody to use, come on, that's a win for the whole field, a win for everybody.
I'm going to go into symptom management as well, which is very important. Some investors have a perception that symptom management is relatively less challenging. I'd like to get Tim's perspective on that, because it could be, maybe more de-risk, which may be an unfair view, but I'd like to get your thoughts on that.
Yeah, it's been changing. I think, you certainly see more appetite now for symptomatic approaches because of the success of Nuplazid and Acadia. They're booking not far off $500 million of sales now annually for that product. You also had the Gopalakrishnan approval for dyskinesia, which has had a challenging commercial launch, but it's improving, and I think people understand maybe why that drug is maybe not having the success that it was initially thought that it would have. I think the other, especially for us in dyskinesia, 5 years ago there was a lot of reformulation of L-DOPA going on, and we know today that these approaches are helping with off time, but they're not solving dyskinesia. I think the investment community very much believes that dyskinesia is a large opportunity, and are starting to invest in it. Other areas, I think some of the side effects of L-DOPA. We shouldn't forget that L-DOPA is the mainstay of therapy. So some of the compulsive disorders, which again are pretty untreated, these are also getting some attention as well.
Sure. Thanks for that. I think we have a few minutes left. I'll start with Spring here, because I'd like to pose a question on what the key challenges or hindrances might be, as a representative of a private company, and then move on to everyone else. Just 30 seconds each. What do you think the key challenges are?
As far as investment perspective, yeah.
On the investment side, how do you generate that spark that gets you the next investment? I guess is the question.
Yeah, we've been fortunate so far. We've had, good enthusiasm for investors and strategic funders, and also non-dilutive grant funding. But I think one of the advantages is that the inflection points are relatively clear in this business. So one of the challenges is that there aren't that many people that invest in early stage, but that's a question that can be asked very much up front. Which stage do you enjoy investing in? And with earlier stages, there's more risk, but there's also the ROI is higher. So, definitely there are players that like to play in that. So that's been, one way that we've overcome that challenge is by bucketing investors into different groups. Another way is, another challenge is often particularly VCs would like some specific data. Pharmas sometimes also would like some specific data. So that can be a challenge because they won't fund that data. They would like to see the results of that data. So that's where the non-dilutive funding has been really helpful for us, particularly, obviously NIH, but also the Fox Foundation. And that gives you back much more than just funding as well. You get feedback from experts at the agency, as well as reviewers. Reviewers will sometimes tell you things that your friends might not. They tend to give you very candid advice and suggestions. So that's been really helpful, a helpful way of overcoming that challenge as well. And then I would say, I guess the last challenge that we've experienced is enthusiasm for PD. And CNS in general tends to wax and wane. We've seen this kind of back and forth pendulum, maybe ongoing. But one nice thing, one advantage that we have, I don't know how well this will work for everybody else, is enthusiasm for mitochondria has been steadily growing. So we've been fortunate in that. But definitely would be sort of nice if enthusiasm for CNS diseases was also steadily growing rather than swinging back and forth. But those are what we've encountered so far.
We're almost out of time here, but I am going to ask the buzzer beater for Marco here. Given you see a lot of things in the space, what's your view on what the key challenges might be to actually sign off on a check?
Yeah, I think a key challenge is just making sure that the projects that we support are ones that are really critical for the applicant to get to that next stage. And that next stage means building a data set, which then makes their work even more attractive. So that greater investment can go in, because I think the Fox Foundation can only sort of go so far with regards to the kind of funding that we do. I used to think that a big challenge that we had was maybe lack of communicating with each other and working on common projects. But just maybe to end on a positive note, I feel that the culture has really changed, and now it's riskier to not be part of these consortiums and riskier to not be talking to your competitors about field enabling questions, because then it's going to be very difficult to move forward.
Thanks, everyone. I'll just turn it back to Murali to have the last word here.
No, I think this has been a great panel, and I'm sure that these topics, we probably can spend another hour and a half talking about this, but really want to take this opportunity to thank each of the panelists for sharing their perspective here. And for those of you who have tuned into this session, we look forward to speaking with you again. Hopefully this has been informative, and feel free to contact us if there are questions or follow-ups that we need to engage further. And with that, we'll close the session today. Thank you all.
Thanks, Murali.
Yeah.
Thanks, everybody.
Thanks very much.
Yeah.